Good morning, everybody. Like Steve said, my name is Shane Corder. I'm the Senior HPC Systems Engineer for the Center for Pediatric Genomic Medicine at Children's Mercy Hospital in Kansas City. And I would really like to offer a beer to anyone who can say that 3 times fast to me later on. So anyway, thanks for coming to the talk. So the center. One thing I'd like to mention. This presentation's a little lacking in some of the areas on the slides because I had a slight computer problem last night and or 2 nights ago and lost pretty much most of it. So anyway, please bear with me. So anyway, the center was established in 2011 by Dr. Stephen Kingsmore, who came from the National Center for Genomic Resources in Santa Fe. He basically came to CMH at the same time CMH was trying to start a world-class research entity. So pretty much things went wonderfully well, and both parties agreed on starting a world-class genome center in Kansas City. So our application focuses mostly on exome sequencing. Our tag scan CLIA test, which is a panel of roughly 500, I believe, diseases, and then our StatSeq, emergency genome sequencing, which is a 50-hour genome. And to my knowledge, we're still the fastest genome sequencing center in the world. Soon, we're going to be working on some other things. I'll talk about that a little later. So Kansas City, this is one of the problems of my presentation here. All the things came in, and it was really nice looking. So where's Kansas, or where's the Genome Center? We're smack dab in the middle in the country in Kansas City. Kansas City is known for, obviously, its barbecue, and if anybody else has anything to say about that, I'll be outside later. We'll talk about that. And we're also known for our fountains and jazz. And just because we're near Kansas, I went ahead and put Wizard of Oz there. So we've had a little bit of acclaim. New York Times front page, along with Mr. Obama and Mr. Romney there. And also in 2012, we were on Time Magazine's top 10 medical breakthroughs at number 7. So those who may not know, a genome is, well, it's your code. It's what makes you you. And you're basically made up of roughly 22,000 genes that code for roughly 100,000 proteins. And that equals to about 6.4 billion letters, the A's, C's, T's, and G's. And if you're a really good typer for 50 years at 8 hours a day, you'd have a stack of paper roughly about that tall. So there's a lot of letters. Another problem with my slide here. Gattaca. I'm sure some of you know Gattaca, which is a great sci-fi movie, one of my faves, about designer babies, the future, basically ridding the world of all health problems, and also giving you that blue-eyed baby, or that blonde or red-haired baby, or that 6'2" linebacker, or 7 linebacker, or whatever. This is not what we do. We do not, we're not in the business of making designer babies. We want to help children. We want to cure them, or at least make their lives much, much better. So as far as the HPC side of things goes, we run a relatively modest cluster of 40 nodes, about 600 to 700 cores. I've lost count recently, honestly. And roughly about 1 petabyte of Isilon storage, and then we have a second tier with the SGI and spectrologic solution. We deal a lot with trying to find out what we can do with the data and where to store it, and we're a relatively new center, so our storage hasn't quite exploded as of yet, but in the near future, we're definitely expecting a massive influx of data with several partnerships that we're doing around the world. So in the past, when you take your child or even you go to the doctor, you basically have all these tests run, and there's not always a diagnosis. I like to say this: 5 years with no molecular diagnosis is more 5 years with no diagnosis, because all of these tests and the $23,000 in cost to you didn't really get you anywhere, besides a lot of stress. So there's roughly 4,100 genetic diseases. Actually, that number has gone up quite a bit since the writing of this. I think it goes up to about 2 to 3 a week, I believe. And those are the #1 cause of infant death, #1 cause of NICU death, and the leading cause of PICU death. A few case studies for you. So CMH 487, the mother was screened at 16 weeks with elevated levels of alpha beta proteins, and the fetal MRIs showed some serious defects on the baby. So after the delivery at the Maternal Fetal Health Center at CMH, which is pretty new, the baby was admitted to the NICU with a ruptured, the ophemocele, which is basically ruptured intestines. After about 2 months, the baby was diagnosed with acute liver failure, and the parents were told that it was pretty dire. They were basically told that the baby was probably going to die. So in steps the Genome Center. The doctors didn't know what to do, so they sent us the sample. After nomination, blood sample was taken, samples to the Genome Center, and these are actually pretty close to the actual time it takes for us to do our thing. Preparing the DNA for sequencing, and then the 24-hour genome sequencing from our Illumina sequencers, which are quite amazing pieces of technology. So there's roughly 120 billion nucleotides sequenced. Then it's sent to these handsome gentlemen here. And there are roughly 2 billion or 2.8 billion nucleotides genotyped. And variants found in those, rare variants found in those, variants likely to alter proteins, 913, which brings us down to 2 variants out of 341 diseases matching the symptoms. So, which comes out to 2 compound heterozygous variants in the perforin 1. And this is pretty much the point where we hand off all data to the analysis group. And basically comes down to a very, very simple treatment. Corticosteroids and immunoglobulin administered via IV. And now the baby's liver function has returned to normal, baby returned home, with the unfortunate event of we didn't find the actual genetic cause of the anomalies, but the baby is alive. The baby is alive and well. So our goals are really to use this to change the treatment for the disease symptoms. We're actually looking to change that to more of a from symptom to actual molecular problem. So here's another one, another really good one. So the baby came in with hypoglycemia. It was a very, very nasty case of it. I am not a medical guy whatsoever. I am a computer guy, so you'll have to excuse me on this. So here again shows you kind of our filtering process, and this was pretty much a home run. I mean, we have one symptom, we have one variant that it comes down to, and ultimately things like this generally are only cured by doing a full pancreatectomy, which leads to lifelong diabetes and many other problems. But with our test, we were able to find that it was only a subsection of the pancreas. This is not the actual pancreas of the baby, but it gives you the same features. So basically, the baby had a 40% pancreatectomy and now is completely cured. So, good news. These numbers are out of date, unfortunately, because every week we have more and more families coming through. So, this comes to 48% of a diagnosis change, which ultimately leads to a treatment change. The big thing here is the 52%. So you say, 50 or 48%. Well, that's good, but what does it offer to those 52% of those people and those families? Well, if you've ever had a sick child or have ever seen a sick child, or even read about them, you'll definitely see that the family goes through months or years of stress, and ultimately can be futile. Sometimes those babies never get better, and so we at least offer a chance to say goodbye to, and also to remove any additional suffering for the baby. So another happy story here. Let's pick it back up again. So we had a 7-year-old boy come into the hospital, and he and 2 of his siblings were seen for muscle tone, lack of muscle tone, basically. Here is the lovely family, mom and 4 children there. The older one is she seems to be fine. It's just the 3 younger ones that were showing poor muscle tone. So after genome sequencing, or exome sequencing, on these, 2 mutations in the nebulin 1 were found, which they were both inherited from mom and dad. So, and then after simple confirmation, it was confirmed. This is right here is the thing that I would like to I'd like to really stress. If you've ever had a muscle biopsy, or anyone you'll know it is extremely painful and not a very fun process. Imagine you're a child, and that multiply it by 1,000. So that was avoided. So, which makes mom a very, very happy person. So the paradigm shift, the boy that you saw earlier had been seen 35 times, and 7 years with no diagnosis, and a great deal of money. And then the 2 younger siblings, seen once, and it took 2 months to diagnose, and roughly $3,000 in comparison. And ultimately, it was a very, very simple change in treatment. And I believe this is the last one I have. So this one can be pretty rough. So, baby was transfused at birth for severe anemia. The brother died of anemia at 4 days, and then at 3 months had a cardiac arrest. So, and all treatments thus far had been completely ineffective. And again, you'll see that the diagnostic workup was very, very, very expensive. So I'll skip to the meat of this one. He had a stem cell transplant, which only one other person in the world had that at that point. And now he's home and he's safe. So this again, this is kind of again back to the what about the 52% down at 2, 3, and 4. So here's all the good stuff here. We've helped cure diseases. We've seen new and unique diseases that we're characterizing and we're actually keeping track of. The palliative care, like I said before, there's no more invasive testing. There's no more futile treatments. And then this is interesting here, the genetic counseling and recurrence of risk, which basically will allow, if we're doing trio sequencing, we're able to tell those parents, you have X% chance of having another child with this disorder. And then they can make further plans for adoption or whatever else. So, in terms of our 5-year goals here, and these are actually have been expanded. We're looking to do a diagnosis for every baby that comes into Children's Mercy, and ultimately find an experimental plan where there's no standard therapy, and then we would like to be able to do it within a month of every child coming to CMH. Also in the future, we are spinning up an RNA-seq program, which will allow us to look at the underlying protein sequences. For those strikeouts that we may have, where we have no real idea, there's no glaring issues with the actual DNA sequence, we're trying to look further down and into the RNA. We're also looking to spin up a children's cancer pipeline here soon, which will be a terribly difficult and in-depth process, but we're hoping to make at least a little bit of headway on that. So these are some of the amazing people that I work with. Every person here is entirely, it's a necessity to have every single person here. And lastly, these guys here, they're healthy, they're totally fine, which is what I want every kid to have a chance to be. So that's it. Thanks.
Thank you. Questions? Mike.
The Illumina sequencer is pretty much all the compute. Is there any compute after you get those results?
No, the Illumina sequencer doesn't actually do any compute. No, it's the sequencer just basically takes images of the DNA, of the strands of DNA that we put on the flow cells. After it comes off the sequencer is where it goes into the compute cluster and into the storage.
That's at Kansas City?
Yes, that's all within the center. We have all of our own sequencing capabilities and all of our own computational capabilities.
How big is that cluster?
That's a 40-node cluster. 40-node, about 700 core, and total about 2 petabytes of storage.
Okay, Jack, just wanted, before we go to the audience questions, your reaction here.
Well, first of all, yay. As a parent, it's always yay for that. I think what this is showing is the promise that you've been hearing about since the genome was sequenced. People have been saying for a long time, since 2001 and everything was announced that, yay, we've got the genome, we're going to have all these amazing discoveries and cures, that it took a while. I mean, with all science, it takes a while. And now we're starting to see these kinds of personalized treatment plans and diagnoses. And that's what we all really wanted from the very beginning. And I think we're at the really early stages. Starting to talk about RNA, the proteins, I will talk a little bit about some of the complexity that we see and where we're going. And then developing therapeutics. Once you actually have the diagnosis, then if you're starting to see this, you can start to develop therapeutics and then get cures and things for that other 52%.
Yeah.
And so I think this is an incredible example of how the technology is driving that and how computation is coming back in to help make those results useful. And time is critical because it's measured in heartbeats in this case.
Yes.
Chirag, question up by you first.
Thanks.
Yeah, actually, I have 2 questions. First of all, thank you very much for the presentation. I think this is why many of us are in HPC today.
Absolutely.
Excellent.
Thank you.
You. Early in your presentation, you mentioned a 50-hour genome, and then during the discussion of case number 487, you mentioned a 24-hour genome. I just wanted to get some clarification on that.
Sure. So the 50-hour genome is actually from start to diagnosis, basically. That's from the time when the baby's finger's pricked to all the way to the physician sitting down with the family telling them, the outcome. The 24-hour period in that slide was actually the sequencing part of it. So that's when the Illumina sequencer's washing over all of the, all the chemicals onto the flow cell. The rest of the time is in DNA preparation and then compute, and then also the end is the final analysis from the physicians and then our genetic counselors.
And what amount of what you're doing is proprietary, or what would it take for other organizations to be able to do what you guys are doing?
Well, a organization would need a very, very supportive back end, like CMH gives the center, and then obviously time, and then but as far as technology goes, you can go to Illumina today and buy a 2500. And there's plenty of HPC vendors out there that would love to sell you some compute hardware, and storage is pretty much the same. As far as software goes, we've spent a lot of time developing pipelines around open source and publicly available software, and also developing our own filtering software, basically. So, and I do know that in the really near future that it's going to be available for license, and we want to like give this to everybody. We want to work with everybody. We want to pass this around the world. We want to get everybody doing this.
Tom Tabor, did you have a question?
Actually, it was very similar to Mike's. I wanted to first thank you for the presentation with miraculous work you're doing. God bless you. And then also just some questions around the sequencing and availability, et cetera. Thanks, Mike.
Other question? Wait for the mic a second, and we'll be right with you. Okay, we have dueling mics.
Who can get there first?
There you go.
Thank
Lob
you.
it.
If you analyze the genomes of parents in advance of them having children, could you determine the probabilities of genetic disorders?
Yes, yes, that's actually, it's been talked about. It's been talked about, and there's certain things that are spinning up around that. Not with us in particular, but yes, you can.
Okay, thank you.
Any other question? Yep.
So, in the 50-hour timeline, how much time do you spend on assembly of the data from the Illumina, and do you do anything special for assembly of a human genome?
Well, we actually don't do assembly because we have the reference genome. So, but as far as compute time, I believe it's total compute time is roughly I think 18 hours, somewhere around there. Might be a little less.
Another question. Susie.
I'm curious as to how much insurance companies are accepting these procedures and reimbursing for it.
Great
'Cause
question.
It would seem to me that if they would get behind it and reimbursed it, it actually could help drive
Yes.
usage of it. That's kind of I don't mean it to sound crass, but
No.
It could be very helpful.
Yes. It can be helpful for both the medical side and the insurance side, and the parents' side, and family side. I don't believe I can actually speak about that, but it's something that's being talked about.
So for the cases that you showed, though, those were out of pocket?
No, actually, I believe those were helped funded by CMH.
Okay, through some kind of grant.
Yeah.
Okay, so I think we'll stop the questioning now. Thanks again,
Yeah.
Shane.